Today's video is on the types of RNA. So first we're going to have a look at some keywords. So codon. A codon is a sequence of 3 bases that codes for an amino acid. The genome is the complete set of genes in a cell. And the proteome is the full range of proteins produced by the genome, which is our complete set of genes. So first we're going to have a quick overview of RNA. So RNA is a polymer, so a long chain molecule, of mononucleotide units, forming a single strand. It contains oxyribose sugars, unlike DNA, which is a deoxy, and this is because of the differing group on the carbon 2. RNA has a hydroxyl while DNA has hydrogen, and then we have uracil base instead of thymine, and it contains a phosphate group just like DNA, and they are mostly single-stranded. So the first type of RNA we're going to look at is messenger RNA or mRNA. So this is a long strand arranged in a single helix. So as DNA is a double helix, messenger RNA is a single helix. And the base sequence of messenger RNA is determined by the sequence of bases on a length of DNA, and this is determined during transcription in protein synthesis. So the base sequence of our messenger RNA is determined by our base sequence of DNA. And mRNA acts as a template for protein synthesis. And mRNA is able to leave the nucleus via the nuclear pores in our nuclear envelope. So structure versus function of mRNA. So mRNA possesses information in the form of codons. So this is our 3 bases. And this helps our function because these codons determine amino acid sequence. So this is why mRNA is used in protein synthesis, because these codons determine the amino acid sequence so our protein can be made at the ribosome. Also, our mRNA can accurately copy DNA base sequences. So this is it acting as a template. So this means that proteins can be faithfully made. So we can make proteins without too many proteins having mutations. We can have accurately made proteins. Also, mRNA is small enough to fit through nuclear pores in the nuclear membrane. So this allows our genetic code to reach ribosomes. Also, mRNA is easily broken down or degraded, and this is because it is single-stranded. So this is able to stop the production of proteins when they aren't needed. So when we don't need the proteins being made anymore, our mRNA is easily broken down to stop the production of these proteins. And it also means that mutated RNA doesn't last long, meaning it can't do long-lasting damage because it's not producing mutated proteins for a long enough time. So next is transfer RNA. So transfer RNA are relatively small molecules of around 80 base pairs long. So these are smaller than messenger RNA. And transfer RNA is single-stranded chain folded into a clover shape. So even though it may not look single-stranded, this molecule here is a single strand of RNA. It's just folded into a clover shape. And tRNA has an amino acid attachment site, and so you can see this here. And so this allows the tRNA to bind to amino acids to bring them to the ribosomes to form these protein polypeptide chains. And many types of tRNA can bind to a certain amino acid. So one amino acid can have many types of tRNA that can bind to it, but only one amino acid can bind to one tRNA molecule. So if this tRNA molecule here is to bind to an amino acid, this amino acid attachment site is only complementary to one type of amino acid, even though an amino acid can have multiple complementary tRNA. And the tRNA also has an anticodon, and you can see this down here. And this is a sequence of 3 organic bases complementary to a codon on the mRNA. So this anticodon will be complementary to a codon on the mRNA, allowing them to line up to bring these amino acids to the ribosome. So tRNA language. tRNA can speak both DNA and amino acid language, and this is because it carries amino acids. So with this amino acid attachment site, it is able to speak the language of amino acids. And it also has a complementary anticodon to the mRNA. So this allows it to speak the DNA language. So now we're going to have a look at DNA versus mRNA versus tRNA and look at all the differences. So structure. So DNA is a double polynucleotide chain. So it's a double helix made of 2 strands. While mRNA and tRNA are single polynucleotide chains. So they're not in a double helix, they're single strands. And their size, DNA is the largest, then it's mRNA, then it's tRNA, which we saw was a relatively small molecule of around 80 base pairs long. Its shape, so DNA is in a double helix, as we can see here. Our mRNA is a single helix, and our tRNA is a clover shape while it is still single-stranded, just folded. And our sugars, our types of sugars in our chains. DNA has a deoxyribose, and mRNA and tRNA have an oxyribose. The types of organic bases. DNA has adenine, thymine, guanine, and cytosine, while mRNA and tRNA have adenine, uracil instead of thymine, guanine, and cytosine. Its location. DNA is found in the nucleus, and mRNA and tRNA, although manufactured in the nucleus, can be found all throughout the cell. And this is because our mRNA has to reach the ribosomes in the cytoplasm for protein synthesis, and our tRNA also has to reach the ribosomes for protein synthesis. And its quantity. DNA, the quantity is constant throughout all cells, except our gametes, of course, which have half the DNA content of a normal cell. While mRNA and tRNA, the quantity in a cell varies depending on metabolic activity. And this makes sense because depending on the metabolic activity of the cell, different amounts of proteins will need to be constructed, so different amounts of mRNA and tRNA are needed. And then lastly is our stability. DNA is the most stable, then it's tRNA, and then mRNA. Because mRNA is a single strand with all of its bases exposed, it can be easily degraded.